I'm Sunil Agarwal. I'm a physician and researcher who currently affiliated with the New York University Medical Center, where I'm doing my residency final year in physical medicine rehabilitation. So I'm speaking from New York City, and I want to give a talk today on cannabis and the endocannabinoid system. This presentation was largely done recently at the American Academy of Physical Medicine and Rehabilitation meeting. So this is very current material, and has essentially I'm trying to lay some scientific foundation for the medicinal use of cannabis and the science of the endocannabinoid system, so that healthcare providers out there have a better idea why cannabis works the way it does, what sort of evidence base we have around it. And unfortunately, much of this material was not covered in medical residencies or medical education, so there's a lot of catching up that needs to be done. But I had the fortune of, I was fortunate to do 4 years of a PhD in medical geography, where I studied medicinal cannabis and had the opportunity to train in a medical cannabis state, Washington State, which that's where I did my medical training, PhD and resident internship. So I had some exposure to use of cannabis in clinical practice. So that's where I'm going to try to give this talk. And the important thing to know is, in fact, the science of this, despite the fact that we know so much, is still in its infancy because of the lack of robust availability of controlled clinical research, empirical treatment trials, access to cannabis, tested cannabis, analyzed material, and so on. So there's still a lot that needs to be worked out, but some basic principles are there, which certainly can help guide later on research development. So without further ado, let's talk about cannabis and the endocannabinoid system, a concise clinical primer and case series. So I want to declare that I don't have any financial relationships with any interest. I am getting a speaker fee to speak here with you today. The talk will discuss FDA-approved uses of drugs, and that's quite a bit of medical practice today goes on with FDA-unapproved drug uses, off-label, as we say. So this is the outline. We're going to go through a little bit of history, then pharmacokinetics, pharmacodynamics, adverse effects, drug interactions, indications, and case series. This was very close to the outline that I followed in a paper that I recently published in the Clinical Journal of Pain, Volume 29, Number 2, 2013. You can see Cannabinergic Pain Medicine, concise clinical primer, and survey of randomized controlled trial results. You can let the organizers know, and I can send you a copy of that paper. So this was the outline that I used in that paper. So let's start with history. Let's see here. Okay. So the history of cannabis in medicine is quite impressive, actually. It turns out that cannabis was part of the materia medica of numerous medicinal systems throughout history. The earliest documentation we have is from ancient Egypt and Chinese pharmacopoeias, but the Indians and the Middle East all utilized cannabis in medical practice. And then even in more modern medical times, ever since the British physicians were in India in the 1830s studying indigenous medical practices, that's sort of when the boom of cannabis in the West was resurfaced. So until about 1840 to about 1940, there was maybe 1,000 or 2,000 different medicinal preparations sold by major pharmaceutical houses that contained cannabis or were solely cannabis. And you can see an example here of a cannabis bottle that was produced by the Mulford Company, pharmaceutical company in Philadelphia, and it contained cannabis in it, and the treatment was for neuralgia, which is interesting because there's a lot of research nowadays that shows that cannabis is useful for nerve pain. The William Osler, the father of internal medicine, Sir William Osler, Sir Dr. William Osler, he had recommended cannabis as the most superior treatment for migraine headaches in his first textbook of internal medicine and several subsequent editions. There was several, the Royal College of Physicians and Surgeons, the Queen Victoria's personal physician, Dr. Reynolds, had also used cannabis for numerous conditions, and there's quite a bit of interest in the Victorian and early 20th century in this treatment, and unfortunately politics and good old American prejudice has gotten in the way and caused this medicine to sort of fall out of favor. Another way to look at history is by studying the publications in the National Library of Medicine and finding out when cannabis or cannabinoids as a term was used in the overall medical literature. So you can do that search for each year from back as far as 1960 all the way to last year. And that's what this graph is showing, is how many hits you get. And it's quite a large number. As you can see, it's been increasing since the early 90s. But the graph also tells you a different history of kind of the moments in cannabis and cannabinoid science, the important moments. So you can hopefully see my pointer here. But in the mid-1964 is when THC was isolated and one of the active ingredients in cannabis by Raphael Mechoulam in Israel. And after that, there was a pretty significant resurgence of interest in the science of cannabis all throughout the late 60s, early 70s. Around this time, the United States government was conducting a United States panel study of marijuana use, and Presidential Commission. And there was a lot of funding given to researchers and a lot of publications at that time. When the panel recommended that the drug was safe enough to not warrant criminal penalties for use and medical investigation, quite a large multi-volume report that essentially took a scientific angle. The report was recommendations were not implemented, and throughout the late 70s there was a concomitant drop in research publications in this area, up until the early 80s when there was really an absolute dip as throughout the Reagan administration. Bush 1. 1988 represented another milestone in cannabis science. The cannabinoid receptor was first cloned by Dr. Alan Howlett at Washington University in St. Louis and her group. So this was the first time that a specific target for a cannabinoid from the cannabis plant was identified in this case it was a mouse, but later it cloned in the human. Discovered in the human and cloned. And that, for the longest time, people had no idea if there was a specific receptor for cannabinoids. And there was very vague ideas that maybe the cannabinoids dissolved into the lipid bilayer of the cell, and they sort of worked non-specifically. Nobody really knew that there was a specific receptor. In 1992, they discovered, again in Mechoulam's lab in Israel, with the collaborator William Devane, that there was actually an endogenous cannabinoid. If there's a receptor in the body, then certainly the body is making a ligand to bind to that receptor, and that was discovered in '92. Since that time, several other cannabinoids and cannabinoid receptors have been discovered, and there's just been a flurry of interest and research in this area as we discovered how widespread cannabinoid signaling is in the brain, nervous system, and other major physiological function systems, not just in humans but in numerous organisms, all the way down to Hydra. Also, during this late 90s, and as you can see, there was a lot of research, which is also coinciding with the increasing legalization of medicinal use of cannabis in states across the U.S. Now there's 20 of them, but the first state, 96, was California, and that, I think the research also peaked as physicians and patients were reintroduced to legitimate use of cannabis medicine. So there's some other ways to look at these numbers at the bottom here. 2.3 publications a day for the last 20 years. So this is a huge amount of new science, and there's no way we can even begin to summate all of that, but we'll pick some highlights. So another way to look at these numbers, in a literature review that was published in 2009 by Hanus from Israel, he said that at that time he could divide up the literature to 15,000 articles on chemistry and pharmacology of cannabis and cannabinoids, and 2,000 articles on endocannabinoids. Another literature review published in 2010 by Hazekamp showed that from the period of 1975 to 2009, if you want to look at strictly clinical studies, that is, studies using human subjects, 110 controlled clinical studies involving cannabis or cannabinoids had been published. And in total, those 110 controlled studies used 6,100 patients, subjects, and with a wide range of different conditions. And that is an important point to keep in mind, given that the prevailing myth is that there's no research and very few people have been studied, and this number, 6,100, is much larger than many other drugs that are currently in practice, used in practice. The United States government has run a cannabis clinical trial informally for, actually that's the fourth point, but before I go to that, the United States has allowed 33 or so trials of inhaled cannabis with cannabis grown at the federal farm in Mississippi, University of Mississippi, by a grower scientist named Mahmoud Al-Saleh. And that cannabis has been shipped out to various medical centers and studied in controlled clinical trials. A third of them were gold standard designed, and that's gone on for the last 30 or 40 years. They've compared that cannabis with placebos, other drugs, or oral THC in various trials. And then there's been this long-term federal study that's gone on since the mid-70s, when a glaucoma patient sued the federal government because for out of medical necessity, because he found that cannabis eased his eye pressure, which was causing him to go blind. And the federal judge sided with him and forced the government to begin issuing cannabis under an investigational new drug program, compassionate use, to him, and then ultimately several other patients who applied. And formally, this is a clinical study, but there's really been no official longitudinal data collected on those subjects, though some independent researchers have done their own studies of these patients. That program is now closed, but 4 patients still received cannabis from the federal government in that program who are grandfathered in. The science on cannabis has gotten to the point where you can, and cannabinoids, where you can do what are called funnel plots. This is a type of plot that physicians and researchers who study evidence-based medicine can tend to see. This is a form of a systematic review and meta-analysis where you can stack up trials that are looking at similar, interventions, and see if on balance, if you stack up all these trials, whether they support one side or the other. And this shows you that the large diamond at the bottom is a summation of the different weights of the different trials that have been looked at for nausea and vomiting for using cannabinoid medicines. There was 18 studies in this particular meta-analysis, and 13 of them were randomized trials. And randomized trials involving cannabis, and you can see here that there is a statistically significant difference in patient preference for in favor of cannabis or its components versus the standard anti-emetic drug. And it's a very statistically significant p-value less than 0.0001. So essentially, a moment please. Okay, so just to sort of show you that this type of literature exists out there. The American Cannabis clinical trials that I discussed earlier, 33 trials have been, there's been more now, but in one paper I published in the Journal of Opioid Management, I summarized at that point, 2009, that these were the different indications that the American trials had looked at with cannabis: appetite stimulation in healthy volunteers, treatment of HIV neuropathy or other forms of chronic neuropathic pain, spasticity in MS, weight loss and wasting syndromes, elevated eye pressure and glaucoma, asthma, vomiting. These all showed, nearly all of them showed benefit for cannabis. And these various clinical trials also have helped set the stage for the different types of indications that cannabis is being used for in the various states that now have medical cannabis laws on their books. The modern cannabis pharmacopoeia has different cannabinoid medicines available, and this is sort of just gives you an idea of those. I mean, this is just the sort of official FDA cannabinoid medicines, but there really are other types of cannabis medicines that are being used in dispensaries and various local medicine systems. But as far as the FDA goes, it's these 4 THC pills on the left, which is 100% pure THC dissolved in sesame seed oil in a soft gelatin capsule. It has been available since 1985 and now has gone generic. Sazamat or nabilone is another chemical that's very similar to THC, but also been available since 1985. Nausea and vomiting and cancer chemotherapy as its indication. The dronabinol has also received HIV wasting syndrome treatment indication as well. And then skip to the far right, you see the cannabis cigarettes that the Mississippi Farm has given in the research studies that I talked about earlier in a long-term study. And they carry a label that you can see here on the bottom of the screen. 300 cigarettes per can. They average weight per cigarette about 0.847 grams. And herbal cannabis, you can it's all rolled up here, but you can see the green poking out of this cannabis cigarette here. Herbal cannabis has about 460 different chemical constituents in it, about 100 or more than 100 different cannabinoids. And then since 2006, the cannabis extraction called Sativex, but essentially it's a cannabis, is an oral cannabis extract, has been available since 2006 in Canada for nausea, for the treatment of pain and multiple sclerosis. This is essentially a cannabis preparation that is made by growing in a greenhouse in England that a private company runs, growing different cannabis strains and taking the harvest of cannabis that has a high THC and one strain of cannabis that has high CBD, grinding them up and dissolving them in liquid carbon dioxide, and then doing some cleaning up of the process and putting it in a glycerol peppermint flavored under the tongue spray. and it was given the name nabiximols by the International Naming Pharmaceutical Naming Group, but it's essentially cannabis, liquid cannabis, or a hash oil, highly refined preparation. And this company has done quite a number of studies to win approvals in over 22 countries now for this cannabis medicine. So that's what I'm showing you in the middle picture there. It's in phase 3 clinical trials in the U.S., both for cancer pain, and I believe they started also a trial in MS. So now let's talk about pharmacokinetics of cannabis. So, as I was saying, best to think of cannabis as a herbal cannabinoid drug. 6,000 different phytocannabinoids. What are cannabinoids? They're these classical phytocannabinoids. They're technically of the class terpenophenolics. They have 21 carbons. There's other things in cannabis in addition to cannabinoids, like other plants. It has terpenoids, flavonoids, phytosterols. Terpenoids are like pinene, limonene, smaller things that give the aromatic aroma of not just cannabis, but other things in the plant kingdom, like limes and pine trees. Phytosterols are like the things that have sort of like estrogens and things like that plants make. The two important cannabinoids to think about are the THC and CBD, and it's important to know that they both appear in their acid form in cannabis. That's carboxylic acid, COOH, if you remember your organic chemistry. In order to activate cannabinoids, you heat them, and it requires about 5 minutes of heating at 200 to 210 degrees Celsius for optimally maximum decarboxylating. But if your temperature is much higher, like if you find in a flame, just a few seconds are required. There's also science that shows the acid forms have interesting medicinal properties for the immune system, but this is a less well-studied area. Pharmacokinetics continued. Routes of administration for cannabis involve the lungs, the gut, and the skin. So by the lungs, you inhale vaporized or smoked organic plant material. The pharmacokinetics is essentially like an IV bolus. Those of you who are familiar with IV administration in hospital. Passive diffusion occurs from the lungs into the blood. And why it's like a bolus is because you have a rapid onset in seconds to minutes, maximal effect in about half an hour, and then duration about 2 to 3 hours. The PO route, the gut route, you ingest lipophilic, alcoholic, or supercritical fluidic extracts of herbal cannabis. The supercritical fluidic extract is what the Sativex, the spray that I was showing in the prior slide, would qualify as. And by the way, that's a technology that's taken from the perfuming industry, supercritical CO2 fluid extracts. Lipophilic extract means butter or oil, alcohol, just like the classical tinctures that are made, alcohol-based tinctures. People use other types of oil extractions as well. So essentially, the absorption time is more variable when you take it by the gut, depending on stomach content. So onset can occur at 30 minutes to 2 hours, and a longer duration of action and more constant, 5 to 8 hours. There's also sublingual absorption as well. Skin is the topical application. There's topical application by the skin as well. This is a less well-studied area, but we do know cannabinoids absorb into the bloodstream through topical administration. And there's been some research in animal models on the use of cannabinoid creams to treat dermatitis, allergic dermatitis in animals and in rats. Metabolism of cannabinoids, mostly plasma protein bound lipoproteins bind the THC in the plasma, and then they go through these hydroxylation, oxidation, and conjugation processes in the liver, like many other drugs that are metabolized. And these are the different enzyme systems that are utilized. Clearance is pretty rapid. First pass metabolism is more significant with oral administration. That is to say, the liver gets a hold of the cannabinoids first before the brain nervous system does. And then the 11-hydroxy metabolite is formed, which has a higher blood-brain barrier penetrability and is associated with more of a more potent effect. The cannabis is eliminated over several days due to storage in fat tissues. Cannabinoids do distribute in breast milk. It's classified as pregnancy category C, which is the one that says that there's insufficient evidence either for or against its use in pregnancy. and interestingly enough, breast milk itself contains natural cannabinoids that the mother passes on to the infant. Excretion of cannabinoids occurs days to weeks and distributed in your feces. Science of cannabis has gone to the level of the genome. This is just a sort of picture showing you what herbal cannabis looks like, the flowers on the left there, and looking at it under a microscope in the middle picture, you can see those white things with the heads on them. Those are called glandular trichomes, and those are basically the medicinal business end of herbal cannabis. It's in those trichomes, which you can see with the electron microscope in the last picture, and the drawing of the trichome on the bottom there, bottom left. This is where cannabinoids are produced by cannabis, and however which way you can get those cannabinoids into your body through inhalation or extraction, that's where the medicinal end is. And this is some of the circulatory pathways, and we've learned exactly how it's synthesized. The genome of cannabis was sequenced in 2011, so there's a whole lot of rich understanding of this plant now. So now let's look at pharmacodynamics. This is a little bit about how the body, how cannabis works on the body itself. And for that, you have to understand the endocannabinoid signaling system, which is really the reason why there's a learner's high, why osteopathic nuclear treatment works, and why electroacupuncture works. These are some slides I took from a sort of pharma outfit, but they show you here that there are two predominant cannabinoid receptors. There's probably more actually, but these were the first two that were discovered. And you can see here that CB1 is distributed in numerous tissues, including the brain, nervous system, but also in muscle, liver, fat, the GI tract, pancreas. And then CB2, more predominantly found in the immune system, although there are some neuronal distributions as well. These receptors are highly expressed in the brain, and turns out they're very widespread. They fall into the class of receptors called G-protein coupled receptors, which are very important. The most famous G-protein coupled receptor, I think, is the rhodopsin receptor. And that's the one that allows you to see, it allows the light photons in the eye to translate into signals from the optic nerve. But the G-protein coupled receptor is known for its transmembrane loops. There's 7 of them that sneak across the membrane. They have a receptor on the extracellular side that transduces signal into the intracellular side. Then below that, you can see the drawings of the endocannabinoids. Anandamide and 2-arachidonylglycerol are the two predominant endocannabinoids. These are phospholipid-derived metabolites that bind to and activate cannabinoid receptors. And you can see that in the upper left, I've also written that this anandamide, which comes from the Sanskrit word for bliss, that it's also not just an endocannabinoid, it also interacts with other systems in the body, such as the endovannelloid system. It binds to the TRPV1 receptor, a little bit lower affinity, and it binds to this other receptor called PPAR gamma. But the TRPV1 receptor is the one that is responsible for helping you taste hot chili peppers. So it just goes to show that these are not just sort of these are integrated into other systems of the body, these endocannabinoids. And you can see the distribution of tissues again on the right: CNS, GI tract, liver, adipose tissue, muscle, pancreas, and then CNV2, these different immune cells. This is they call them cartoons or crystal structure. There's no crystal structure, but it's kind of showing the different components of the crystal, of the protein structure of the CB1 receptor. On the top is the what you're seeing here are the different transmembrane loops in the middle there. Those are called alpha helices. And then you have these extracellular side and intracellular side above and below. When you activate the extracellular side on top here, it's negatively coupled to amyloid cyclase. That's what the signal that comes down here causes, a negative coupling with amyloid cyclase. Calcium conductance in the neuron is suppressed, and there's an inhibition inward rectifying potassium channels. And this leads to a suppression of excitability of that neuron. In the neuron, again, it's very interesting to note that the mechanism of action of cannabinoids is retrograde, and that's very different than classic neurotransmission, which has always been studied and taught to be anterograde. And that is to say, signals travel from presynaptic to postsynaptic neurons in a studied fashion. Here on the left is the neuromuscular junction, showing the acetylcholine being released and binding to the acetylcholine receptor here, and then being chewed up in the synaptic space there, the cleft. However, the endocannabinoid system goes in the absolute opposite direction, postsynaptic to presynaptic. Endocannabinoids are synthesized on demand from the lipid bilayer of the postsynaptic neuron, and then they are released through some kind of transporter, and then they go and bind to a cannabinoid receptor on the presynaptic terminal, and then there's an inactivation process and a breakdown process of those different endocannabinoids that happens inside the cell. So this system is very unique in nature. There are a few other retrograde presynaptic receptors in dopamine signaling that have been studied, but this is the only signaling system that we know of in human biology which its only mode of signaling in the neurons is retrograde. And what that allows endocannabinoid signaling to do is play an important role in feedback and regulation of neuronal excitability. That is to say, if there's too much signal coming from presynaptic to postsynaptic, the postsynaptic cell is able to send the signal back to the presynaptic neuron and tell it to reduce the signaling of that particular neurotransmitter, let's say glutamate. On the other hand, it can also help to upregulate signaling if there's not enough signaling. So you play a kind of role as a symphony conductor at the synapse. This is a picture of distribution of cannabinoid receptors in the rhesus monkey brain. You can see here that from frontal lobe to the cerebellum, these are sagittal slices that have been taken before the monkey was sacrificed. He was injected with a radiotracer that binds to cannabinoid receptors. The red indicates highly dense tissue distribution, and green less dense but not zero. Black is close to zero. So you can see as we go from frontal lobe and take these sagittal slices that cannabinoid receptors are everywhere in the brain. The cortex, the basal ganglia, the caudate nucleus, here, in the cerebellum here. relatively few in the brain stem. The same experiment has been done in the human brain. Here you have uptake of this radio tracer in a human who was not sacrificed but put under a PET MRI scanner. You can see a similar distribution of these receptors in the human brain. This was done at NIH. These curves on the bottom are binding curves that are able to prove that, in fact, this is the cannabinoid receptor that is being targeted. This was published in the Proceedings of the National Academy of Sciences in 2007. The authors noted that CB1 receptor is one of the most abundant G-protein coupled receptors in the CNS, rivaling the abundance of benzodiazepine, striatal dopamine, and ionotropic glutamate receptors. It is tenfold higher than that of opioid receptors. So this is a significant receptor system in the brain, in CNS. Because of the widespread distribution of cannabinoid receptors in various tissues, you can imagine that they physiologically play important roles. They help to reestablish cellular homeostasis, essentially, and have been shown to affect a large number of physiological processes, feeding and energy balance, GI function, metabolism, the perception of pain, motor control, posture, learning and memory, and emotions, immune responses, inflammatory responses, cardiovascular function, reproduction, bone formation. So it's quite a widespread system, and studying it and understanding it really can help to guide a whole new understanding of human physiology. And I recommend an article in the Journal of the American Osteopathic Association called the Endocannabinoid System and Osteopathic Perspective by John McPartland. It really helps to put that sort of whole system medicine context to understand this system's functions, and that article does a great job of that. I think it's available freely. So because we have this understanding of cannabinoids now, we can understand how it is that cannabis helps to stop nausea and vomiting in cancer patients, which is an indication that's been known about since the 70s, widespread known about in the United States. But for the longest time, people had no idea exactly how it worked. And now that we have this understanding of cannabinoid science, we can say, oh yeah, well there's cannabinoid type 1 receptors and neurons in the medullary nucleus solitarius in this brain stem, which is, we know, as part of the circuit, the emesis brainstem neural circuit. The area is called the chemoreceptor trigger zone. Cannabinoid inhibition of receptors in this area, of the serotonergic receptors, increase inhibitory control, which may be why we get less nausea and vomiting. So this explains this inhibition is through that same retrograde signaling function that I was describing earlier. Essentially, it happens with endocannabinoids, but also if you give the patient cannabinoids, you can upregulate that inhibition process and downregulate the activity of the part of the brain that makes you feel nauseous and want to vomit. There's other pathways too, but this research was done in the shrew, lesser shrew type of lab animal. So anyway, that's part of how cannabinoid science can help you to understand its function in medicine, cannabis' function in medicine. As far as pain goes, cannabinoid receptors localize to all kinds of areas in the anatomical distribution of tissues that are important for pain processing. One of the most important ones to note is the periaqueductal gray, which is an important area in the brain for descending pain processing. That is to say, when pain is how the brain sort of shuts down pain For example, if you are stubbed your toe but you're being chased by a saber-toothed tiger, you really aren't able to address the pain at the time, so your body has a mechanism of suppressing those type of pain signals. And that area that's responsible for that in the brain is full of cannabinoid receptors. They're also in the thalamus, the dorsal root ganglion, amygdala, and cortex, which are other important areas for pain processing. So the immune cells, as we discussed, the CB2 receptors, they are involved in modulating the release of chemokines and cytokines, which can induce inflammation and thereby also cause pain. So it turns out that that was just sort of static anatomy, but also you can show that the cannabinoid system is part of the way that animals respond to the induction of pain through nerve injury. At least 9 different animal models of this is quite torturous on the animals, I must say, but this data is here, so we should look at it. That there's surgically induced traumatic nerve or nervous system injuries have been studied, and these different 9 models: constriction injuries, partial nerve ligation of the sciatic or saphenous nerves, spinal nerve ligation of the L5 nerve, spared nerve injury, spinal cord injury, tibial nerve injury, or streptozocin-induced injury, which is a model for diabetic neuropathy. And all of these models, this was an article published in Neurotherapeutics in 2009, we've shown that cannabinoids are involved in the body's response to these painful injuries. So, for example, in the chronic constriction injury of the infraorbital nerve, cannabinoid 1 receptors are then upregulated in the trigeminal caudate nucleus after this injury has occurred. They're actually upregulated both in the nucleus on the same side that the nerve is cut and on the opposite side. CB2 receptors are increased in the dorsal horn of the spinal cord at the level of L5 after you cut the L5 spinal nerve. After you cut the saphenous nerve partially, you have an upregulation of mu-opioid, CB1, and CB2 protein levels. That's found in both in numerous tissues. In the hind paw, both on the ipsilateral and contralateral side, and the dorsal root ganglion, and the ipsilateral and contralateral lumbar spinal cord, which is the origin of the saphenous nerve. And you see these changes 1 to 7 days after you do this injury to the nerve. If you cut the tibial nerve, which is also in the lower extremity, you have an increase of CB1 messenger RNA in the opposite side of the brain, which is the pain relay station, the thalamus, just 1 day after the injury. The spinal cord injury model has shown that there is mechanical allodynia, that is pain hypersensitivity, decreases when you give cannabinoid medicines to a spinal cord injured animal. Cannabinoid agonists called WIN. But there's no decrease in the effectiveness of that medication over time on the pain, which is different from morphine.